Today's video is going to be all about proteins. Just like carbohydrates previously, many of you are probably already familiar with the term, or at least in some context. Any type of packaging on food, such as nutritional labeling, will display a section allocated to proteins, which would probably be measured in grams per serving. Along with that, if you're athletically inclined, you might associate proteins with products that may help you build muscle. While these definitions of proteins are correct, we will begin to see that proteins are involved in or play a role in practically every single biological process and each and every living organism that we know of. So you may now have asked, what are they exactly? The answer would be that they're biomolecules, just like carbohydrates. Proteins are actually very immense biomolecules, as it falls under the molecular family of macromolecules. Here on the screen, we can see all the different levels of complexity in the protein structure. We can now see the quaternary structural form of a chaperonin protein, in which we can see all the abstract figures that are created by the protein structure. This protein is roughly 800,000 times the mass of a hydrogen atom. By that rationale, we can conclude that it's going to contain tens of thousands of atoms, making it very much a macromolecule. Something to be careful of, however, is that while these proteins are colossal on a molecular scale, the largest protein that we know of, Titin, is only 1 micrometer in length. That makes it substantially larger than our previously displayed chaperonin protein. A micrometer is still 1,000th of a millimeter, so even the largest proteins remain very microscopic. Another way to conceptualize proteins is by what they are made of. For example, some proteins can be made of a single chain of a substance called amino acids. Other proteins, such as chaperonin, titin, and hemoglobin, are made of multiple chains of amino acids. Further on, we'll get into the properties and reactions of particular amino acids, but for now, you can simply imagine them as the building blocks for proteins. If you watched the previous video, you may have inferred that the amino acids in these chains are monomers for the polymers of what's known as a polypeptide chain. So each chain of these amino acids is known as a polypeptide. For smaller proteins that only consist of one chain of amino acids, that polypeptide is the protein itself. Whether the protein has one or more polypeptides, when the monomer amino acids bond with each other to form the polymer polypeptides, they bend and warp, resulting in the abstract shapes that we see in the structure of proteins. So you can imagine the hemoglobin protein here to be comprised of 4 different polypeptides, as they warp, bend, and have a conformation that forms their shape. This in turn is what gives proteins their power. As I mentioned, proteins are involved in almost every single biological function. They play a structural and mechanical role. For example, in an isotonic muscle contraction, such as flexing your bicep, the proteins actin and myosin can be found interacting with each other in a boat rowing-like formation to contract the muscle. Proteins can also act as enzymes, which will be gone into further depth in the future. Enzymes can help catalyze chemical reactions. They are a key component in biochemical reactions that take place in biological systems. They can be involved with our immune systems or sensory fibers in the nervous system, for example. These are just a few of the millions of scenarios in which proteins are often used, proving that proteins are incredibly important. With that out of the way, we can now dig deeper into the building blocks of proteins, which are amino acids. So what we can now see here is a typical structure of an amino acid. We can easily see some of the elements within this acid, such as oxygen, hydrogen, carbon, and nitrogen. Then, bonded to the carbon atom in the center is this R. You may be confused as to what that is because none of the elements on the periodic table of elements have a symbol of just R. That's because this is not an element. The R refers to sort of a placeholder for a side chain, which would then differentiate from the common amino acids. Here on the screen, we see all 20 of the amino acids that can make up proteins. We can see that their basic structures are all identical. The only thing that is different amongst them is their R group, or side chain. Now we can easily see which segments of the amino acids are the side chains. For now, We don't necessarily need to know the biochemistry of each side chain in too much detail, but we can see that it's these different side chains that give us the diversity of all the proteins that we know of in biological systems. Whether it's their structure, role, or specialties, they are all dependent on the variation of these amino acids. Now, if we go back to our chaperonin protein, which is involved with helping other proteins to get their shape, it chaperones the protein's folding process. We can only imagine the complexity that lies within this single protein. It's such a vast machine with multiple abilities, but it forms naturally in biological systems. And as we explore more and more of biology, we will continue to see these fascinating proteins, which are all really incredible systems that astonish our imaginations. We will continue to go over all these vastly unique biomolecules of science to further strengthen our base of knowledge and understanding. But for now, I thank you all for watching.